also oral and maxillofacial pathology department at Tehran University of Medical Sciences School of Dentistry. And also I would like to thank Appraise to Raise team for technically support this meeting. It's a great honor for me to have Dr. Shama Virani from WHO Institute International Agency for Cancer Research on Cancer. And also we have Dr. Sophie Rousseaux from Grenoble University, but unfortunately, Dr. Sophie let me know a few days ago that one of her relatives passed away in an accident, and unfortunately, she has to attend funeral ceremony today. But she kindly recorded her lecture and sent it to me. And at the beginning of this webinar, we will share her video. Let me share some information about Dr. Sophie Rousseaux with you. She is a member of epigenetics and cell signaling department at Institute for the Advanced Biosciences in Grenoble University. And the aim of this department is discovering the molecular mechanism governing the post-mitotic reprogramming of the male genome. Also, she kindly had a collaboration with us at Oral and Maxillofacial Department at Tehran University of Medical Sciences for about more than 3 years, and we published our joint research about 1 or 2 months ago in British Journal of Cancer. And I think she will share this project in her lecture with you. Thank you, Shama, for being with us. But let me share some notes with you at this part of the webinar. As Dr. Sophie well experienced, that it is very important to have a very collaborative team to conduct a cancer research, especially people or experts who are well familiar with bioinformatics, and also clinicians. And it is very important to make a bridge between clinicians and who are well familiar with bioinformatics, because all information in clinics are based on findings and big data gathered from genetics information. And it was, as Dr. Sophie well experienced, it was a great research we conducted in collaboration with the Grenoble University. And I would like to invite you all again that share your questions with us, and I will send them all to Dr. Rousseaux. Let's go to the second part of the webinar that Dr. Shama Virani kindly accepted my invitation to this webinar. She is a cancer epidemiologist in the genetic epidemiology group at the International Agency for Research on Cancer in WHO in France. She's focused on using genetic epidemiology to understand cancer inequities in incidence and survival globally. Dr. Virani, thank you again for accepting my invitation, and we are all ears for your presentation.
Great, thank you. I will go ahead and share my screen.
Okay.
Can you see that okay?
Yes, great.
Great. Okay, so first I would just like to thank Dr. Amini Shakib for inviting me here today I would like to give a little bit of an overview on all of the head and neck cancer research that we've done over the last, well, I would say decade or so in the genomic epidemiology branch, and see where there may be opportunities for collaboration, as you know, this is very important, especially when it comes to head and neck cancer, as I hope I can show you. So we've been doing head and neck cancer studies for a very long time. The first studies was probably back in 1998 with the Central European study and with the South American study. So both of these studies were created in different parts of the world to try to understand the differential risk profiles of the acknowledged and accepted risk factors of tobacco use and alcohol use, as well as the potential role of viruses in the oral cavity and other head and neck cancer sites. Following that, in 2002, the ARCADE study was launched, and this was a study for alcohol-related cancers and genetic susceptibility in Europe. This is where we started getting into more looking at genetic susceptibilities using genotyping data, and I will talk a little bit more about that in a second. In 2011, the Interchange study was launched. This was a study looking at the International Cancer of Head and Neck Genetics and Environment study. This was focused in South America. In 2016, we started to take an approach of using more consortium style approaches, where we have many different studies coming under an umbrella of targeted and focused research. And this is also around the time where we were more focused on looking at prognosis from head and neck cancers. So this was the Voyager Consortium focused in North America and in Europe, looking at the role of germline and somatic DNA mutations in oral and oropharyngeal cancers. And just recently in 2019, the Headspace study was launched, funded through the European Commission. This looked at translational studies of head and neck cancer in South America and in Europe. I just wanted to take a moment to highlight the different populations that have been included in all of these studies. And I just touched on the main studies. There's a lot of smaller ones as well, and the importance of their contribution. So of course, there are populations globally that are generally represented, maybe even overrepresented in cancer genomics work. But it's really important, especially, well, for head and neck cancer, but really any kind of cancer genomics study, to get as many diverse populations included as you can. And so the countries highlighted in red here are populations that are generally represented, and the countries that are highlighted in blue are countries that have generally underrepresented populations in cancer genomic studies. And we are always trying to look for centers to contribute to our studies in different parts of the world. And I will keep coming back to this concept because it really is a driving factor in how we do this type of research in our branch. So, you know, we've been doing these studies for a long time, and the biorepository at IARC for head and neck samples specifically has grown quite a bit. If we just look at samples that were contributed to our biorepository between 2002 and 2020, you can see that from a few of the studies here, we've amassed a large number of samples, and they are a wide variety of samples as well. So we have fresh frozen or formalin fixed paraffin embedded tumor samples. We have blood and their blood products. We have normal tissue, normal adjacent tissue. And now we are starting to increase our collection for oropharynxes and buccal cells as well. Now, if we take all of the patients that we've collected throughout all of the studies, and this is what we consider our head and neck cancer group, we can have a quick look at their demographics and see that they really do represent what we would expect when we think of a head and neck cancer population. So we see that we have a 3-to-1 ratio of men to women. 75% of our patients are male. Most of them fall in between 41 to 80 years of age. When we look at the number of patients across the different subsites, we have quite a large number. So if we look specifically for samples that came from patients who had oral cavity tumors, we have about 5,000 samples. We have, and these are tumor samples. We have about 3,000 larynx samples, 740 hypopharynx samples, and 4,000 oropharynx samples. So it's important to think about the role that these samples play, because as I will talk about in some later slides, the potential for their use is now becoming more and more relevant as we improve the technology for genomics research. Of course, the tobacco and alcohol status is mostly for current users. And when we look at the geographic distribution across the North American, South American, and European populations, we can see that there is generally a 1-to-1 ratio. I will point out that in the European ancestry group, we do have a lot more controls. This is because people of the European ancestry, those in Western and Northern Europe and those in North America, are very highly represented in cancer genomic studies. And so many consortia around the world have controls that we can use in our study as well. So that's where the high number of controls is coming from. Now, in the past, we really have focused on very specific types of data, as you can see here. So we have genotyping data on about 22,000 subjects. And this data was generated from 2 large-scale genotyping efforts that were conducted in 2011 and subsequently in 2013. Following this was we have a large number of samples with HPV serology data. So what is HPV serology data? It's a multiplex assay that is able to detect human antibody levels against 8 high-risk HPV types. And you can see the HPV types listed here. They're all high-risk types. And they look at antibody levels against all of the proteins across the genome. And then we have a small number of samples that have whole exome sequencing. So you can see that there's very specific data types that we have accumulated in the past. And so I want to spend a little bit of time talking about why it's so important to have these pieces of information. So in terms of HPV serology, we use this as our measure of dictating whether or not a sample is HPV positive or negative. Now, obviously, there's a lot of different measures we can use. We can use P16, we can use HPV DNA, HPV RNA-ish, but we like to use HPV serology. And I will talk about why that is. It started with the International HPV Prevalence Study. So this was a study that wanted to assess the prevalence of HPV in Europe. So that was through the ARCAGE study that I mentioned earlier. In the US, so this was done through the CHANCE study, which is based in North Carolina, and in Brazil. So this was done through the GenCapo study in Sao Paulo. So we were able to collect patients from these different areas and then assess the prevalence of HPV16 among head and neck cancer cases from these regions. Now, this paper was published in 2017. The data was collected and analyzed probably in 2014, 2015. So at that point in time, we were starting to understand the importance of HPV as a risk factor for head and neck cancer. And the evidence that I'm going to show you now really formed the basis of all of the evidence we've generated of why we know HPV is such a strong risk factor for oropharyngeal cancer. So of course, the highest prevalence was detected in the oropharyngeal tumors as compared to the other head and neck sites. But what is interesting is that if you look across the different geographic regions, so you look across the US, Europe, and Brazil, the prevalence still differs quite a lot. So in the US, it was about 60%, in Europe, it was about 30%, and in Brazil, it was only 4%. This really underlines the importance of considering different populations around the world, because the circulation of HPV16 in the population differs based on behaviors of certain populations, risk factor profiles, etc. Now, it's important to look at survival differences by HPV status. And of course, none of this is new to any of us in the audience here, I'm sure. But when stratifying by HPV-related versus HPV-unrelated oropharynx tumors, we see there is a clear difference in survival probabilities for those that have HPV-related cancers being favorable. And in other head and neck cancers, you do see a difference in the survival curves, but the extent is much lower. And again, when you look across the different geographic regions, you see that even these differences in survival tend to change based on, of course, the HPV prevalence and the number of HPV-associated oropharyngeal tumors that are found in these regions. So you see the strongest association with survival in the US, strong association in Europe, and then a slightly strong association in Brazil, but with the few cases of HPV-positive tumors there, it's difficult to go much further than the estimates we see here. Now, of course, it's important to think about the other epidemiologic factors that contribute to survival. So we know that HPV HPV infection is really important. And whether you are measuring P16 or HPV DNA status, we know that if you're positive for both, you tend to have a better prognosis, so a lower risk of death compared to those that are negative. We know that a risk of death increases with age. We are also have indications that women have better prognosis than men do. There are other factors to take into account as well. So of course, with stage, if you are diagnosed at a increasing stage, going from 1 to 4, your probability of survival decreases, your hazard of death increases at the later stage you get diagnosed. And of course, if you're a regular tobacco user, your hazard of death increases as well. Let's go back to HPV for a second and think about how we're defining HPV status in these types of studies. So let's take two ways of measuring it. We have HPV16 DNA and we have P16 status. Both are heavily used in the clinic. These are definitely good ways of assessing HPV16 activity. And P16 is what's being used in de-escalation trials to stratify HPV positive versus HPV negative patients. So if we look at just HPV16 DNA, and if you're positive for HPV16 DNA, you see that there is a lower risk of death, a lower hazard of death compared to those who are HPV negative. Again, this falls in line with what we would expect. And separately, if you looked at P16 status, you would see there's also a lower hazard of death if you're P16 positive versus P16 negative. But what happens when you use both of these markers together? So if you're negative for both, or if you're positive for both, you see the association that you would expect. So if you're P16 positive and you're HPV16 DNA positive, you know that you have a better prognosis than those who are negative for both. But what happens if both of these markers are discrepant? If you're either P16 negative, HPV DNA positive, or if you're P16 positive, but HPV DNA negative, what do we do with that information? We can see here that when we start to look at the hazard ratios for the groups that have these discordant results, we are not able to see the trends that we would expect. But they're both measuring HPV, so why aren't we able to see what we would expect? The definition of HPV is really important because we know that HPV can interact with other known risk factors. And I'll just show the example of smoking here, where you can see that for the survival curve in green, which is for never smokers and those who are HPV positive, they have the best prognosis. Those who are smokers and HPV negative have the worst prognosis, again, what we would expect. But in the middle, if you have patients who are never smokers and HPV positive, which is this blue line, or smokers but HPV negative, it's really difficult to start to elucidate some of the associations here, which one has the better prognosis because of this interaction. It's also important to think about how we define HPV because we anticipate an increased burden of HPV-driven oropharynx tumors in the coming decades. Now we know that HPV-associated oropharynx tumors is increasing in many different countries. And in countries that have cervical cancer under control, oropharynx tumors are becoming the primary HPV-related cancer. But again, this varies geographically. And this is a point that I keep coming back to and why it's so important to look at different regions of the world when assessing HPV-driven oropharynx tumors. So what is the best way to measure HPV? Well, you're looking at the HPV16 genome here, and you can see the nucleotides in the black circle here. You see the proteins that are associated with HPV. So you have E6 and E7, which are the oncogenic proteins, and then all of the other ones here. One way that we can measure this is by looking at the DNA of the virus, presence or absence of HPV DNA. Now, this can tell us if the DNA is there, but it doesn't tell you if it's active, if it's being transcribed. And so that is a limitation because we know that HPV viruses can lay dormant for many years before being activated. And for oncogenic effects to be exerted, the HPV virus has to not only be a persistent infection, present for many years, but it also needs to be an actively persistent infection. So we need to know something about the activity of the DNA. The gold standard is really to use HPV E6 or E7 expression. And this is good because it tells you not only that the DNA is there, but also that you're getting, it's active. It is actively transcribing and expressing the oncogenic proteins that will eventually cause our downstream oncogenic effects. However, you need to get tumor tissue for this. So it's a bit invasive. It's possible to do if you have a good study setup, but it is a little bit difficult to, constantly be collecting tumor tissue. The other way that we can do this is by measuring P16, which is something that we do in the clinic a lot in many different centers. But as we all know, P16 is an indirect measure. We know that E7 binds to retinoblastoma. We know that this increases P16 expression, but there are other cellular mechanisms that increase P16 expression besides HPV16 proteins. So we start to get a little bit of misclassification there when we only rely on P16. One way that we can measure HPV16 effectively is through the serum. So there's this HPV antibody assay that was developed by our collaborators at TKFZ in Heidelberg. So HPV16 E6 serology, which is what this assay measures, antibodies in the host against the proteins that are oncogenic. It appears to be an extremely specific and sensitive marker for oropharynx tumors. And I'll show you some of the data that supports this in a second. We know that it has really high specificity. So in many studies where thousands of samples were measured, less than 1% of controls are HPV16 E6 positive. The nice thing about this is that you can use this assay in a potentially, in a very small amount of blood. So we're talking 50 microliters of serum. The first study that looked at this as a biomarker was the European Prospective Investigation into Cancer Nutrition, or the EPIC study. The centers that contributed to this are shown in the map on the right. About 400,000 participants gave a blood sample at some point. Some of them went on to later develop oropharynx tumors. So the HPV serologic biomarkers were evaluated in head and neck cancer cases and controls. The time of the blood draw to the time of their cancer diagnosis on average was about 6.5 years. Now, there's a couple of things that really stood out in the results of this initial study. The first was in the controls. So patients subjects who did not go on to develop cancer. There were about 1,600 controls that were measured, and you can see that the antibody against HPV16 E6 was present in less than 1% of them. They were present in other sites of head and neck cancer, but this biomarker was most prevalent in oropharynx tumors. And when we looked at the odds ratio of developing oropharynx tumors when you have this biomarker, we see that it's the strongest for oropharynx tumors. When survival was assessed using only this biomarker as a stratifying factor, you can see that the HPV16 E6 positive, the biomarker positive cases, had a much higher prognosis than those that were biomarker negative. Now, this biomarker was subsequently validated in the PLCO consortium. And again, we see what we saw before, where in the controls, a large number of controls, there was a very small proportion of those positive for the biomarker. The patients who ended up developing oropharynx tumors had the highest proportion of positivity. And when we looked at the risk of developing these different cancer sites, we saw that the risk was highest in oropharynx tumors. The nice thing about the PLCO consortium was that they were able to collect serial blood samples over time. So we were the authors were able to measure this biomarker in many blood samples before diagnosis. What they found was that if you look at years from blood draw to diagnosis, so that's on the x-axis here, where on the right is year 0 where diagnosis occurred. And you compare the E6 fluorescent units, which is on the y-axis here, the cutoff is at 1,000. So if you're higher than 1,000, you're considered positive. If it's lower than 1,000, you're considered negative. What they found was patients who had high positivity, so they were considered positive up here, stayed positive throughout the entire time until they reached diagnosis. And those who were negative stayed negative until diagnosis. There were a few people who seroconverted, as you can see here. So some people do increase over time, but very, very few. This work was actually validated again in another consortia, because this was starting to look like something that could be very, very useful in terms of screening. And so for HPV-C3 consortium, this was also looked at. And what was found was that it wasn't only 12 years prior to diagnosis, but all up to 26 years prior to diagnosis, where positive biomarker levels could be detected before the diagnosis of the tumor. Now, naturally, you see these results and you think, well, can this biomarker be used to screen for preclinical oropharynx cancers? You know, the specificity is very high and the sensitivity is also very high. But we should remember that oropharynx tumors are rare, and we don't know what to do with a precursor lesion. If we did know what a precursor lesion even looked like, and if we knew what to do once we found a precursor lesion, or if there was even a therapeutic vaccine available, so after infection, able to interrupt the process of the HPV proteins affecting the host genome, if we had a tool to do that, then we can use this information. But as of now, we have this idea that this biomarker can be detected many years prior to diagnosis, but we don't have anything to do about it. So this is another line of research that's going on in our branch of finding another biomarker that can be used in collaboration with this biomarker closer to diagnosis that would help us indicate, you know, when people should be screened for this cancer if it's a good idea. Nonetheless, when it comes to research, the genomics research, the epi research, this is why the HPV16 E6 antibody is used to stratify HPV positive versus HPV negative patients. Another big component of the work that I've shown you is in germline genetics. And so we spent a lot of time focusing on the susceptibility to oral and oropharyngeal cancers. And we have quite a lot of samples that have genotyping data for this reason. So I'll talk a little bit about the work that's been published recently. In 2016, we ran a genotyping study. So Dr. Lesueur, who was working in the branch at the time, conducted the GWAS study for oral and oropharyngeal tumors. She identified new susceptibility loci, which was in and of itself quite exciting. But what was more exciting was the fact that this HLA locus in 6p21 was significantly associated with a reduced risk of HPV-associated oropharynx tumors. Why is this area so interesting? The human leukocyte antigen region is what is responsible for detecting foreign pathogens and eliciting an immune response to those pathogens. The more variable this region is, the better your likelihood of detecting foreign pathogens. And what we found was that this particular haplotype, this class 2 haplotype, if you have this, you have a reduced risk of HPV-associated oropharynx tumors, presumably because this is very specifically focused on HPV16 E6. Now, that was the first kind of foray into this work where we identified this haplotype, and then we found that this haplotype was previously identified to protect against cervical cancer as well. So it's very specific to HPV16. And it implied a role for this haplotype, a biological role, that if you have this haplotype, you're able to eradicate HPV-infected cells before you go on to initiate the process of tumorigenesis. Now, because this was such an interesting finding, another colleague of mine, Dr. Ida Ferrero, decided to look very deeply into this region. So she conducted fine mapping of the histocompatibility complex, which is where the HLA region is located. And she was able to identify 2 independent protective variants. So if you look at the Manhattan plot here, this is only in chromosome 6, where the 6p, which I showed you earlier. And now we're looking very deeply into this region. And there's 2 peaks here, one here and one here. Now, because of linkage disequilibrium, some of these can travel together, you know, when you pass on your genes. So we were looking for independent markers. And the ones that were best associated after fine mapping was this SNP here, and also this SNP here. So what does that mean? Well, the SNP that was found, the 2 SNPs that were found were both associated with the reduced risk of HPV-driven oropharynx tumors. So in and of itself, that was already interesting. But we had all of the serology data. So the serology data is, if you think about what it means, they're antibody levels. So it gives you an indication of your immune response. And when we looked at the different antibody levels against these HPV proteins, we found that one of the germline variants that was identified was associated with a decreased level of antibodies against E6. And the other one was associated with a decreased level of antibodies against L1. Now, for those of you that are familiar with HPV genetics, we know that E6 is the oncogenic protein. So if you have high levels of that, you're going to likely induce tumorigenic effects at some point downstream. But L1, if you have L1 antibodies, it simply is an indication of cumulative exposure to HPV. You don't necessarily have to get cancer from this, but you're just exposed to HPV. And we are exposed all the time to HPV16. In fact, our bodies are quite good at clearing HPV16. It's a very small proportion of those who are infected that go on to develop HPV-related cancers. So taking this picture into account, we have two germline variants that are associated with your humoral response, your antibody levels, and also that are associated with the decreased risk of HPV-driven oropharynx tumors. This work was just published this year in Nature Communications. Okay, so I talked about the data that's been generated in the past, but we currently are in the midst of generating a lot of data from the two consortia that I mentioned on the first slide. So the Voyager Consortium and the Headspace Consortium, and then some other projects that have come out from that. So I just want to give you a general overview of what's coming. So from the Headspace Consortium, which is focused in South America and Europe, we are developing ctDNA methylation in liquid biopsies. So this is going to be done in about 500 patients. When we find a somatic profile, we essentially want to interrogate the liquid biopsies to see if we can capture any of that somatic profile in circulating DNA. We have 800 tumor normal pairs in which we'll be conducting whole exome sequencing and tumor methylation. And in about half of those, we will be conducting gene expression. In the Voyager Consortium, which is focused in North America and in Europe, we're running a targeted cancer panel on over 2,000 tumors. So this is already larger than the TCGA, which is again, very exciting. The targeted cancer panel was developed by Dr. Neil Hayes, who was heavily involved in TCGA. And it says 800 cancer genes, but in fact, it's well over 1,000 very well-known cancer genes that we're looking for somatic mutations in. The nice thing about this assay is that it also sequences a large number of infections, something that is of a personal interest of mine and something that I have recently been started working a lot on. So for every single tumor out of these 2,000 tumors that's HPV positive, their HPV genomes will also be fully sequenced at very high depth. Another area of my work is the genotyping aspect. So, my interest is in incorporating diverse populations because the germline genetics, the germline architecture does have an influence in your risk to different cancers. And so since our last genotyping effort, which was back in 2013, a lot of the centers that we work with have been collecting more cases. And so we have a large enough number now to run another round of genotyping. So we do have germline DNA that's going to be run for genotyping from Europe, from the Middle East, from Tehran University, in fact, with Dr. Kazem Zendele, in South America and in North America. And then, of course, we'll be running HPV serology on all of our oropharynx tumors. And last but most certainly not least, the Mutagrouse project is a CRUK funded project that supports whole genome sequencing across 5,000 tumors, a subset of which will be head and neck cancer tumors. And the goal of this is to identify mutation signatures that can be associated with specific exposures. Okay, so how are we going to be using the genotyping data that we're generating at the moment? Well, we have previous genotyping efforts, and we will have now more. So we're going to combine all of the genotyping data that we have from the past and from that's being generated now. And if we combine all of the cases and controls, we will have a total of 14,500 cases and approximately 26,000 controls. Now, this does not include potentially public available data sets that we can use, such as the UK Biobank or ALSPAC, and we will be including those as well. The inclusion of the larynx subsite is really important here. So for some reason, larynx, the focus for head and neck cancer in a lot of the studies that have been done in GEM have been focused on oral cavity and oropharynx. And the larynx, generally speaking, is quite an understudied subsite when it comes to genomics of head and neck cancer. To date, I could only find one GWAS on the larynx subsite, and that was in Chinese populations. So we're really looking forward to elucidating some of the genomic susceptibility loci directly related to this subsite. Of course, the diverse populations, as I mentioned earlier, will be including the regions that are listed here. And the human leukocyte antigen region story that I just mentioned earlier, we have these specific germline variants that decrease risk of HPV-driven tumors, but the samples that those came from were all European samples, European ancestry. And it's possible, just because of the sheer variability of this region, that if you look in a different ancestry, you'll be able to find different germline variants that are associated rather than these. So we're going to be looking specifically at this region in the diverse populations to see, are we seeing the same variants or are we going to be seeing different ones? I mentioned that there was a pretty vast amount of genomics data that was being generated. So the natural question is, how do we plan to use it? Well, there's three main components of how we plan to use that data. The first is through somatic characterization. The second is through genomics of disease progression. And the third is through clinical translation. Over time, as we've conducted studies over the years in the branch, there has been a general focus on prognosis and on understanding how the disease progresses to a place where you end up having poor prognosis. One big focus of ours is to assess late-stage diagnosis. This is in terms of like sociological measures, but genomics is what we do best. And so we're also asking that question from a genomics perspective. So for the somatic characterization, we will be describing mutation patterns in relevant subsets. So things like by stage, by the different subsites, and also biologically by HPV status, et cetera. We will also molecularly define to the tumor subsets with measures that we've developed in our group. So things like tumor burden, polygenic risk scores, driver genes. This is all work that's currently being done in the Mutograph study as well. In terms of disease progression, we have a lot of omics data that's being generated. We have mutation data, we have expression data, we have methylation data. So we need to integrate all of that information using supervised and unsupervised clustering methods and look at their association with recurrence and prognosis. And what we hope to do is, once we have this idea of what is important, to match their profiles in liquid biopsies of the same patient, pre-treatment, and then hopefully post-treatment as well. Clinical translation is something that is always an end goal. We want to be able to translate our findings. And so we'll be developing prognostic models for 5-year overall survival, incorporating the epidemiologic, clinical pathologic data with the genomic data, using decision tree algorithms for survival probability, probably with neural network approaches. And we'll also be able to ask new questions like, what is the effect of the exposures mediated through somatic alterations? So what does that mean? How do the smoking related mutations, we know there's certain mutations related to smoking, how do those contribute to progression and overall survival? Okay, so we are also assessing exposures, and I touched on this earlier when I mentioned the Mutagraphs project. So through the Mutagraphs project, we have about like 450 tumors that are going to have whole genome sequencing available, and we will be assessing the data for specific signatures. There are specific signatures that exist that we know are associated with tobacco, and there's an ApoBec signature that is associated with viral exposures, so HPV. But it's difficult to tease out, especially if there's a lot of different exposures happening in the same person. One way to kind of get around this is by doing a very careful sample selection. So you can see here in this 2x2 table that we have patients who obviously are smokers and drinkers, which is the largest number. But we have been able to identify patients that have never drank but do smoke, that don't smoke but do drink, and then have neither exposure. And by setting up the study like this, we're able to kind of tease apart some of the signatures that have been found in the past and see if there's anything new that we're missing in terms of mutational signatures that could be playing a role in head and neck cancer. So that one is focused on new signatures, and also tobacco and alcohol. But there's also the story of the human papillomavirus, and this is work that I'm conducting with my postdoc. What I'm interested in is looking specifically at tumor host dynamics. So understanding how the HPV viral genome interacts with the human genome and what that means for overall survival. We know this is important because, as you can see from the survival curve, this is from a paper that I've published a few years ago now, this green line is the survival curve for patients who are HPV positive but integration negative. We know that the HPV viral genome integrates into the host genome, and we have some evidence now, including this, that that integration is bad for prognosis. This red line here are those who are HPV negative. The middle blue line shows patients who are HPV positive and integration positive. So their tumors are integrated with HPV DNA. And you can see the difference in the survival curves between those that are integrated and not integrated. So there's something going on here about how the HPV viral genome interacts with the host genome and what that means for downstream mechanisms. The other important thing to think about is that HPV16 has been around for a very, very long time. And as humans evolved and came out of Africa 70,000 kiloyears ago, the HPV viral genome was actually still there. So as people separated into the populations that we see now, the HPV virus traveled with them and they co-evolved with their populations. And what we're finding now is that HPV16 genomes have variants just like the human genome does, that helps them to survive, that helps them to be persistent in their infection and evade the immune system. And I'll just give you an example. There are certain sublineages, and you can see the sublineages here. There's A, B, C, and D, and then within that, there's other phylogenetic arms. There are some of these, let's take A4, for example. So the A4 sublineage of the HPV16 genome, which basically has a couple of variants, tends to persist longer in Asian Americans than in any other population. So the genetic ancestry was really important because this sublineage evolved with Asian Americans over time. And so again, knowing in different parts of the world what kind of sublineage you're dealing with can also become important. It's not just about HPV16 anymore. It's about the variant inside HPV16 that could be playing a role in prognosis. Okay, so just to end on the last few slides, I just want to reiterate that it's really important to have as much representation as possible in these types of studies. These are large-scale international studies. Everybody here knows that the head and neck cancer sites are very diverse, and so you do need large numbers, and for that you need collaboration. But it's not just about numbers at the end of the day. We could work in North America and get numbers, and that's fine, but you need diverse populations because it is becoming more and more clear that your germline genetics, the DNA you're born with, has an impact on your susceptibility to getting these diseases. So the more representation we have of different populations around the world, the better we'll be able to understand that dynamic. And there's always opportunities for capacity building. You know, I can say, so I coordinate the Voyager and the Headspace Consortium, and just the amount of learning that goes in all the different directions, from the centers to us and to the centers across each other. I mean, there is just so much to be learned, and I have, really appreciated that rich exchange of ideas from so many groups around the world. It's been really nice to be part of something like at that scale. So there is a future impact that we hope to achieve with everything that I've talked about here. We are developing a very rich data set, and the goal is to eventually harmonize all of this data. I mean, we have genomic data, we have extensive risk factor data, we have clinical pathological data, and putting that all together in a way that's easily used is also something that I'm currently working on and trying to get funding for. If you look at the specific types of data we have, these offer a really nice opportunity to do some biomarker work for early detection and for prognosis. And considering the different sample types we have, we can now start to ask questions that we potentially couldn't have been able to ask before. I mentioned capacity building. So collaborative centers are always generating their own data for their studies. And we have our interests on a global scale where we want to compare across populations. So we're hoping that our resource with that interest in mind can also help enhance the center's research by providing additional data. Eventually, we are hoping that this will be used as a global resource. So I have a plan of developing a data hub at IARC, and this is under construction at the moment, to have a way of sharing this data with external scientists where they can come forward with a research proposal, get access to this different data, and be able to ask questions that are more relevant to their context or their setting. So this is something that we're also working on and hope to see happen very soon. It takes a village, as we all know. All of these projects would not be possible without the help and dedication of so many people across so many different places. And so I would have to just take a moment and just go through the acknowledgements here from all the different projects and say thank you to them as well for being able to put together all of this work so that I can share it with you today. Thank you so much.
Thank you, Dr. Virani, for your great, fascinating presentation and great conclusion at the end of your presentation. It seems that we need a big data set to conduct great research on head and neck cancer, and you are doing great research in your center because you are in collaboration with a lot of centers around the world. Thank you again. We have several questions from our audience, and let me share the first question with you. In clinics, clinicians think that there is difference between oral cancer and oropharyngeal cancers, especially regarding HPV infection. What's your idea about it? I saw that in most research you consider both of them as a single cancer, oral and oropharyngeal cancer. But as you have seen in recent published American Joint Committee on Cancer, there is a difference between oral cancer and oropharyngeal cancer, and HPV-associated oropharyngeal cancer have a good prognosis, but it is not the same as oral cancer. What do you think about it?
So in all of our research that we do treat oropharyngeal cancer and oral cancer separately, oral cavity cancer. So this is coming down to, I think, semantics. I'm currently contributing to the WHO Oral Cancer Handbook that we're writing right now with experts from around the world. And believe it or not, one of the main questions has been, is it cancer or is it cavity? Oral cancer or oral cavity? Because in fact, oral cancer, the term oral cancer is everything, includes the oropharynx tumors. But oral cavity cancer and oropharynx tumors are two different sites in oral cancer. So it does come down to how we talk about it. That's really important. And what we do in our research is we stick to the ICD-10 codes because we know that it's base of tongue, we know that it's tonsil, we know the specific sites that are associated with HPV. And so when we are looking at by subsite, we do consider the ICD-10 code and make sure that we're capturing the oropharynx versus the oral cavity.
Okay, sure. And we have the participant POCOF3 raised hand, and would you like to turn on your microphone and video to share your questions? I don't have your name. Would you please introduce yourself and let me ask you to unmute. Did you raise your hand accidentally or you have a question? Okay, let me share another question with Dr. Shama and we get back to the audience. Is there any association between HPV infection and smoking, or they are two different etiologic factors for oral and oropharyngeal cancers?
So we're starting to see now that there's a possibility for there to be interactive effects between smoking and HPV. As I showed in the figure earlier, we know that the survival curves are different if you have both exposures. And so what that tells us is that there is likely, if you're only HPV positive, you know that you have a better prognosis. If you are a smoker, you know you have poor prognosis. But once you have both of those exposures in whatever combination, that the survival curves are in the middle of those two. Now, trying to extract how multiplicative those exposures will be is difficult because of measurement bias. So again, the issue of how we're measuring HPV, but also a lot of the smoking data that we have is self-reported. And unless you start bringing in cotinine measurements or something that can really truly tell you about a smoking exposure, you will always have this measurement bias that you with self-reported data that will always be there. And that has been the biggest, I would say, difficulty in trying to really quantify what the multiplicative effect of smoking and HPV is.
Great. You will explain about it is very important how to measure HPV infection, and you will explain that HPV16 E6 antibody would be a reliable biomarker. But would you please compare it with P16 as immunohistochemical marker? You know that P16 is routinely used in labs to screen HPV-associated oral or oropharyngeal cancer. Which one would be more reliable to be used in labs?
So I think based on the evidence, it's quite clear that HPV serology can be superior because we know there are other mechanisms that influence P16 expression. However, running that assay in a clinical setting is difficult. And in fact, what we're doing right now through the Headspace Consortium is we're pilot testing using an ELISA kit to be able to run the serology assay in different clinical settings around the world. So we're looking in centers in South America, and we're looking at centers in Europe, trying to see if it's feasible to even do that. If it's feasible, then we have some way of now saying, please stop using, well, you can, please, in addition to P16, please think about using HPV serology, and here's how you can do it. Because the Luminex assay that's being conducted in the lab is very expensive, and it's just not feasible in a clinical setting. But ELISA is much cheaper, and that's the way we're trying to go through it, pilot testing it in a clinical setting.
Great. Okay. It means that it depends on the feasibility of the test. As you may know, that IHC study is more feasible at the labs, and it seems serological test might not be feasible enough to be used in routine practice in histopathological laboratories.
Well, they might be. So again, we're pilot testing ELISA kits to do the same thing. And ELISA kits are much cheaper, and they can do more of a high throughput approach because you can do many samples at the same time. So stay tuned. We will let you know what we find.
Yes, I hope so. Okay. And you will explain about your collaboration with Dr. Zendedel. I think he's I think president of cancer research in Tehran University of Medical Sciences and in Cancer Institute Hospital. Could you please more explain about this collaboration and how other researchers in Iran or other countries in Middle East can be in touch with you and make collaboration?
Sure. So I think Dr. Paul Brennan, who has the PI for a lot of these studies and who's been doing this for many years, has been working with the Golistan cohort for a very long time. And through that, we've met many amazing researchers, one of which was Dr. Kazem. And so just recently, when we were putting together our Headspace Consortium, he mentioned that he has, I think it's about 600 case control pairs for head and neck cancer. And so when I was trying to find funding for the genotyping study, I said, well, would you like to be part of that? I think the biggest thing about collaboration is just being able to talk to people and say, what is your interest? Are you able to contribute anything in terms of samples? What would you like in return? You know, this sort of thing, and having that dialogue going. So now we have that collaboration with the genotyping study. He is also starting a prospective recruitment for our head and neck studies, which is great news. And we have a member of his team, Elmira Ibrahimi, who is now at our institution at IARC doing her PhD with us. So it's been a really nice evolving of the collaboration, and we always welcome more collaborators.
Okay, thank you very much. And it was a great presentation. And I would like to invite all audience to follow the page, because the webinar has been recorded, and you can be in touch with our lecturers. And as Dr. Shama well explained, that you can be in touch with her and IARC and WHO to conduct your research in collaboration with the researchers at the WHO. Thank you very much again, Dr. Shama. And I would like to invite my colleagues to share the certificates of the lecturers. First, I would like to express my, my condolences to Dr. Sophie. She's not at the meeting at the present time because she lost one of her relatives, but we will send her certificate to her via email. And the next one, please. Thank you very much, Dr. Shama Virani, for your great collaboration with us. I'm sure this is a beginning for all collaboration, and we will continue, go ahead this contribution. And I hope to be in touch with you in near future. Thank you very much. And I would like to invite all the audience to turn on their webcam to have a group photo for our archive, and we will save it in our web page and share it with you. Thank you very much. Thank you. I know you might not be at a appropriate, convenient situation, but if you can, please turn on your webcam to have a group photo. Thank you. We can wait for a few seconds to have more audience. Okay, hello Amir, hello Aaron, hello Fatemeh. Great, great. I would like to ask my colleagues to take photos and share it in the web page. Okay, thank you very much. Thank you very much, all audience. I do appreciate your participant. Okay, thank you, Ali. Great. And hope to be in touch with you in near future.
Great.
Have a nice day.
Thank you so much, everyone. Thank you. Bye-bye.
Have a nice day. Bye-bye.